A cell-mediated immune response begins with the activation of a small number of T cells by a specific antigen. Once a T cell has been activated, it undergoes clonal selection. Remember, clonal selection is the process where lymphocytes proliferate or divide several times and then differentiate, become more specialized cells. And they do that in response to a specific antigen. The result of clonal selection is that we have formed a clone of cells that can recognize the same antigen as that original lymphocyte. Some of the cells of T cell clones become effector cells, while effector cells are going to cause an action to occur, while other cells become memory cells. So the effector cells of the T cell clone carry out the immune response that's going to eventually result in the elimination of the intruder, the pathogen. At any given time, most of our T cells are inactive. Antigen receptors on the surface of the T cell, simply called T cell receptors, recognize and bind to specific foreign antigen fragments. Remember, the antigen fragments the antigens are going to get broken down by those antigen presenting cells, and then that antigen and MHC complex is going to be put out on the cell surface so that the T cells can recognize it. So there are millions of different T cells, but each one has its own unique receptors that recognize a specific antigen MHC complex. So specific T cells respond to specific antigen fragments. Antigen recognition also involves other receptors on the cells. So T cells have receptors that are CD4 or CD8 proteins. These are referred to as co-receptors. Okay? So there are T cell receptors that are going to bind to the antigen fragments on the MHC complex, on those antigen presenting cells. But the other receptors on the T cells, the CD4s and the CD8s, are co-receptors. So antigen recognition by the T cell receptors and a CD4 or CD8 receptor is the first signal in activating that T cell. Most T cells that display the CD4 develop into helper T cells. These helper T cells, of course, are going to recognize the antigen fragments at the surface of the antigen presenting cell, and then the helper T cell becomes activated. Once that helper T cell becomes activated, it undergoes clonal selection. And remember, clonal selection is simply you're making clones or copies of that specific type of cell because it was activated in response to that specific antigen. So the result of that clonal selection is the formation of helpers or clones of those helper T cells. So now we've got helper T cells, and we also make some memory T cells from that clone. So if you look at this graphic, you have a specific antigen and MHC complex that's going to activate a specific T cell that has specific receptors for that antigen, for that complex. So that specific T cell becomes activated. It clones itself, proliferates, and differentiates, makes more, and specializes. So copies of that particular cell become helper T cells that are going to be responsible for eliminating the pathogen, and memory T cells that are cells that have a long lifespan that, remember, they're clones of this cell, so they have the same type of receptors. So the next time that particular pathogen with that particular antigen is in the body, then those memory T cells can proliferate and differentiate and become those active T cells of the same exact type. So now we have memory T cells that have been formed that are going to be able to respond the next time that particular antigen is present in the body. But those active helper T cells are going to start secreting a variety of cytokines. So cytokines are small proteins that enhance the activation and proliferation of T cells, B cells, and natural killer cells. So the helper T cells are going to help the immune response by secreting chemicals that cause more cells to be activated and more cells to be made. Most T cells that have that CD8 receptor develop into cytotoxic T cells. Cytotoxic cells recognize foreign antigen molecules on the surface of cells that have been infected by microbes. They also recognize some tumor cells and cells from tissue transplants. Once activated, the cytotoxic T cell undergoes clonal selection. So they've been activated. Specific T cells that are cytotoxic with specific receptors have been activated by specific binding of antigens, just like we saw with the CD4 cells. So the CD8 receptors are bound by those antigens, and once those cells are turned on, then they're going to clone themselves, resulting in more cytotoxic T cells that consist of active cytotoxic T cells and memory cytotoxic T cells. So the active cytotoxic T cells are going to attack other body cells that have been infected with that antigen. The memory cells do the same thing as the memory cells we saw for the CD4 cells. They're not going to attack body cells. Instead, they're going to be available to proliferate and differentiate into more of the same type of cytotoxic T cells and more cytotoxic memory cells if the same antigen enters the body at another time. So cytotoxic T cells are the true soldiers that are going to fight these battles. Okay, so they do battle with the foreign invaders in the cell-mediated immune response. They leave the secondary lymphatic organs and tissues and migrate out to destroy infected tissues and cancer cells and transplanted cells. Cytotoxic cells recognize and attach to target cells, and then they deliver a lethal hit that kills that target cell. Cytotoxic T cells have receptors for specific and very particular microbes, and so they only kill the target body cells that are infected with one particular type of microbe. So how do they do that? Well, cytotoxic cells can release granzymes, which are protein-digesting enzymes that trigger apoptosis or cell death. Once the cell is destroyed, then all of the microbes that were killed are going to be destroyed by phagocytosis. Cytotoxic T cells also infect body cells and release two proteins from granules that they store inside their cells. So those granules can contain perforins or granulosin. So perforins are going to insert into the plasma membrane of the target cell and create channels in the membrane. So extracellular fluid can rush into that target cell and the cell ends up bursting. Other granules in the cytotoxic T cells release granulysin, which enters through those channels that the perforins created and destroys the microbes by creating holes in their plasma membranes. So the cytotoxic T cells can destroy target cells by releasing these toxic chemicals. Another one is called lymphotoxin. Toxins created by lymphocytes. So these enzymes cause the target cell's DNA to fragment, and of course that leads to cell death. Another really cool thing that the cytotoxic T cells do is after they have destroyed a cell, they will detach from it and go out and seek out even more targets to destroy. So they go out and hunt for even more invaders.